Hello, viewers. Recently, a team of Bangladeshi scientists at the Child Health Research Foundation have successfully managed to complete the genome sequencing of SARS-CoV-2, that is a coronavirus, in Bangladesh. The team of genetic sequencing was led by a very promising microbiologist, Dr. Shejuti Saha. We have her with us today to know all about their journey and beyond. We also have with us the executive director, Dr. Shomi Saha with us, who also guided the whole team. First of all, we'd like to know from you, Dr. Shejuti, what you have done so far in this pandemic situation in Bangladesh.
Oh, thanks for the question. So, our journey actually started back in March. So, we know the first 3 cases were detected in Bangladesh on the 8th of March. And since then, we have been trying to understand what is the best way to serve the country, to serve the government. On the 29th of March, the Child Health Research Foundation was enlisted as an organization to provide diagnostic testing services for COVID-19 to the Bangladeshi population. Since then we have received tremendous amount of help from DG Health Services and of course the government of Bangladesh. So as soon as we started testing and looking at all the global reports and realizing that there are groups around the world who are sequencing the genome of SARS-CoV-2, we have also been thinking how we should be sequencing the virus too, the viruses that are circulating in Bangladesh. So we know so far more than 24,000 genomes have already been sequenced. So right then we actually started to gather resources that are required to sequence. So before this, we have sequenced several different bacteria. We have sequenced other respiratory viruses like influenza virus, RSV. So we had some, I would say, practice of how to deal with nasopharyngeal swab samples or oropharyngeal swab samples and look for viruses and sequence them. So that's how it all started. And then in recent weeks, when we were seeing that many genomes have been sequenced in India, even Pakistan sequenced a genome, Nepal sequenced a genome, it almost became this stubbornness, this challenge that we also have to put Bangladesh on the map. And we are hoping that now that we have shown it is possible to sequence such organisms in Bangladesh, we're hoping that there will be more groups who will also be sequencing as we go along.
Dr. Saha, you were here in Dhaka to lead the whole team of Child Health Research Foundations. Shejuti was working from afar from London. Can you please tell us what was about leading the team?
It is really amazing because if I go a little back, then when I really doing my work or my research, the PCR was not even discovered on those days. So now, forget about the PCR sequencing. So now the new generation, they are really doing sequencing, and I really just facilitated their work. And I know maybe what need to be sequenced, but they know how to sequence and they know what to do. So this young generation, I say that my group is very at different stage. For example, I have members who are coming back after doing PhD, and I have members who are doing PhD, ongoing PhD, and I'm working with the team. I have members who are on the way to go for PhD, going to abroad. So all them are together. They have really contributing a lot, and it is amazing that this young team, not maybe the next generation, the very new generation, they really have done all these things. So I'm very impressed, and then I'm very proud of my team.
Thank you. Shejuti, if you tell us about this genetic sequencing, what is the importance of this sequencing?
Thank you. So all living organisms or organisms have either DNA or RNA. This is basically the building block of every organism. If we can sequence that genetic material, we can kind of understand what the organism looks like, the specialties or the phenotype or the peculiarities of the organism. So coronavirus, that is the cause of this pandemic, is an RNA virus, which means it has RNA inside it. And sequencing the RNA means, RNA is made up of 4 letters, A, C, G, U. Sequencing means understanding the sequence of those 4 letters, how they're arranged. So if we go back to the beginning, we know that China first declared about this mysterious pneumonia-like illness on the 31st of December 2019. All we knew then was that it's a mysterious pneumonia-like illness. We didn't have much more information. Then we realized it was some kind of a beta coronavirus, but we did not have much specifics. Then on the 9th of January, the first genome sequence was announced of this virus that was causing that pneumonia-like illness. And that is when we realized it was SARS-CoV-2, novel coronavirus, right? So that is the first reason why sequencing was important then. Then, we have a lot of questions about where did this virus come from? Is it from like snakes, this, that bird, this bird? There were a lot of controversy. But because we were able to sequence specifically SARS-CoV-2, and we know sequences of the coronaviruses in bats, we know now very confidently we can say that this coronavirus came from bats. There were probably another middle organism through which it passed to come to humans. But again, sequencing helped us for that. The primary test that we do for diagnosis of COVID-19 is a PCR test. We have been able to design PCR tests because we sequenced SARS-CoV-2. Otherwise, we would not have such a good, reliable test. So these are all the reasons that in general, knowing the sequence helps. In addition, we know that there are many vaccines that are in trial. The first vaccine that went on trial is an RNA vaccine, which means it was designed based on the sequence of the virus. Now, why is it important to sequence the specific viruses that have come into Bangladesh? All viruses change. When viruses change, we call that mutation. Every virus has a different rate of change. Coronaviruses also change. For example, influenza virus, it changes so rapidly that every year we need a new vaccine. Coronaviruses do not change as rapidly. We think on average it changes about twice a month, which means we see about 2 mutations per virus per month on average. So as these vaccines are being made, the question has become, is this virus going to change so much that the vaccine will not work in certain places of the world? Or when the vaccine is being designed, should all the genome sequences or a representative genome sequences from the entire world should be taken into account? So there were questions about how the virus is changing or the genome is changing in different parts of the world. When we see genome sequences and knowing the rate of change of this of the genome sequence or the rate of mutation in this virus, when we sequence viruses, we can kind of predict when the virus came into the country, what does it look like now, where did it come from, and what do we predict, where is it gonna go? In addition, we also know that there was not only one introduction of coronavirus into the country, there were probably multiple introductions. Probably someone came from UK, someone came from Italy, someone came from China. So understanding that point of introduction also gives us an idea of what are the circulating viruses, how should we tackle them, and what are the epicenters of the infection, and what do we predict for the future? Plus, will the vaccines that are being designed work in Bangladesh?
I wanted to know very specifically that there is a large volume of genome library globally. So what will be the impact of sequencing from Bangladesh? Will it have any impact in the vaccine development, or they will include our strain?
That's a really important question. So this is something that actually ties back to what we have seen historically. We have seen that there are other vaccines that have been made in the past using just genomic data from strains from, it's usually in the case of bacteria, strains from Europe, Australia, or America that did not take into account the genomes of the circulating bacteria in South Asia. And that's mainly because genomes were not available. However, now that when we look at those bacterial genomes, we realize that bacteria over here, not only people, bacteria here are also different. And researchers who developed the vaccines do regret that they did not have that data then. So absolutely, every genome sequence, it is our responsibility that when we sequence a genome, we make it open access. It goes into international platforms like GISAID, NextStrain, where all our genomes are publicly available, and researchers working in vaccine development have access to the data, so they can also monitor the changes and include that in their analysis, research, and development.
Okay. Dr. Shah, I would like to know that how did you develop your team for this sequencing? Because not many labs in Bangladesh are doing genome sequencing. So you have built your capacity and you have that manpower.
I think that is the way Challenges Foundation is moving. And Challenges Foundation is focusing, and then we really, I think the only way, the reason the Challenges Foundation can get this success, because Challenges Foundation does basic science, but maybe unlike many other basic scientists, we link it very much with the public health. So that's the reason we thought that at this stage, maybe it is very important to have this get sequenced, and that is the way we'll be giving a very big impact on the public health. And that is the reason that one strain sequenced in Bangladesh. Think about this. There's so many great things are happening in this country, but I think that created a huge attraction from the people of the country, and even the people who never heard about sequencing, but they wanted to understand what's really going on, because it has a huge public health implication. So maybe we are not doing many great things what the people, other scientists are doing in the country, but those are very hard basic science, maybe publishing in Lancet or maybe in Nature or maybe Science. But we really wanted to do something, merge this basic science with the public health. And that is really giving a different type of, I would say, different type of impact of the country. And that more really helps the people of the country and the country itself.
So far, how many bacteria did you sequenced? What is the number? And
so, if you consider this, I mean, you were talking about what kind of bacteria and how many bacteria we have sequenced, or you were talking about the coronavirus?
Coronavirus sample.
Okay, so the coronavirus we really sequenced only one. So Shijiti will be moving with the scientific, and her team will be talking about more science and gene. I really translate in a different way. I translate in a way, this is the first time that it is being sequenced in the country, and it is sequenced by Bangladesh people or the young scientists of Bangladesh. And also it is uploaded on the website, putting Bangladesh in the name. So that's one thing that every time people will be opening that website, they'll be seeing that Bangladesh has got something and it is coming from Bangladesh. That's very important for us. I think we all should feel proud, not the scientists only, as a Bangladeshi citizen, we should be feeling proud about this. And I do so in fact.
And Shijiti, how long it takes to complete the sequencing of one sample?
Right. Thank you. That's a really good question. How long does it take to complete sequencing of one viral genome? It actually completely depends on what kind of resources you have, what sequencing machine you have. So before we can load a sample into a sequencer, we have to prepare a library to sequence of that viral genome, and that takes about 2 to 3 days. And a sequencer machine can take anywhere between 17 hours to 48 hours, again, depending on the machine. So we have a very small machine. It's called an iSeq 100. It is the smallest Illumina platform sequencing machine that is there. And it takes 17 hours to sequence one virus. However, in bigger machines, you can do hundreds of viruses in 24 hours. So that's really dependent on what kind of infrastructure and resources you have.
So in Bangladesh, do we have a sequencing machine in other labs?
Thank you. Yes, to my knowledge, there are 15 or 16 sequencers currently in Bangladesh in different research facilities.
Okay. So, Dr. Shahab, can we make a collaborative effort to engage them for sequencing more virus strain?
I believe they are already in the process of doing it. Is only maybe we just did it little ahead of time. That's the reason we got uploaded it. But this is really bringing a very big momentum in the country to do more and more sequencing. I really came to know that there is a group in our university from where I am. They are really planning to sequence thousands of strains from Bangladesh. So I believe that this is a momentum, and no matter who is doing, all together in Bangladesh, there will be a large number of strains will be sequenced very soon.
I would like to know what were the main constraints for doing this sequencing from your lab?
Sure. So I told you that it was the end of March when we were really trying hard to figure out how to sequence. The first constraint was getting all the reagents and the supplies together. It's already quite difficult to get everything into Bangladesh or our lab. On top of that, the COVID-19 situation throughout the world basically made shipment really slow. So we had to figure out different ways of getting things into our lab. Like I communicated with my collaborators in Canada, in San Francisco. I was talking to people in Singapore and kind of trying to see who can send me a little bit of reagent from here or there. Even if companies could not send me, people were sending me reagents from their own lab, their own stock. So while I'm really, really grateful for friends and collaborators to help us, at the same time, that was definitely challenging. And then just getting everything ready, up and running. So it took us about a month to get everything together and working in the lab. So our lab, we have about 80 people now actually in CHRF who are completely devoted to the COVID-19 response, and 17 to 22 people now actually work in the lab, handling samples, testing samples. So because our first priority is always giving service to patients, we always knew that no matter how much research we want to do, how much we want to sequence, it cannot impact patient service. So the team, my sequencing team, they were doing their normal 8-hour shifts. So we run 24 hours, but people have 8-hour shifts. So we have 3 shifts per day. So my sequencing team were basically working 8 hours. And after they were done with that, they would start their sequencing work. So that was definitely challenging. And thirdly, which was quite a learning lesson, is, as you know, I am locked out of Bangladesh. I am not there physically. So being able to manage and communicate in real time was definitely a learning step. There are cameras everywhere, speakers and microphones everywhere. They're pipetting and asking, Apu, is this okay? Is that okay? What do we do next? But the collaboration was real time, and I will say it was difficult, but it was amazing, also a learning process.
Okay, great. We know that training in genome sequencing may prevent the next pandemic or the flattening the curve. It may help a lot. So since you were pioneer in Bangladesh sequencing this coronavirus, will you be planning to train other centers or other people to sequence this coronavirus in Bangladesh?
Thank you for that question. Absolutely. So again, going back to what Baba was saying, the lesson to take from all of it, or what we are proud of the most, is that the sample was collected in Bangladesh from a Bangladeshi patient, and it was sequenced in Bangladesh. And that is what we have to remember, because traditionally, historically, we've always sent our samples outside the country. And whenever samples leave the country, we lose data ownership. So I think knowing that we can do this in Bangladesh is the first step. We have optimized the protocols, and it is our ethical responsibility to ensure everything is public. All protocols are public. Anybody should be able to use them, and our ethical responsibility to provide with any technical help that is required. There are many groups in the country some with sequencers, some like us with technical expertise. We have the technical expertise, but we have a small sequencer. I think all groups have their weaknesses and strengths, and this is our opportunity to bring all our strengths together and move forward in a collaborative way.
Thank you. Dr. Shaha, your organization is one of the organizations that are doing testing the coronavirus sample in Bangladesh. If there is any other support you need from the government or any other institutes from Bangladesh?
I think we are getting support from the government because we started with the kit from our own kit, but then finally we moved to the kit from the government because at this point we do not have the kit. We ordered, but it's really not arrived yet. But otherwise, we have all other supports. I mean, the kit is not the only thing that you need. You need to extract. You need all the appliances, in fact. You need all the infrastructure and the machineries. But all those things, we are having it. And we really do not need help at this point, that type of help. But I think we are getting, help we are getting, we are very much in touch with the DG Health. For example, the ADG, the Assistant Director General, Professor Nasima, she is very much with us, and then we really text with each other. And we are also in touch with IDCR, and then we also text with the director even she is in the meeting, and we really be in touch with them. Any problem, or we need anything, we just communicate with them and we get immediate response. So we need resources also, and we are supported by some other collaborators. So all together, we are still okay, but definitely we'll be reaching if we need anything. But otherwise, these are all the equipment-wise and other things. But in addition, all supports we are getting from the DG Health and IDCR. And IDCR, I'm saying once and again, because when we started, IDCR was the main group. So we still continue to share the data with them. And all together, both the groups are helping us a lot, in fact, honestly.
You were testing the coronavirus samples throughout the country from different referral centers. So what do you think about the situation of Bangladesh when we get the curve flattening here?
That's the $1 million question. That is the answer, maybe question. I can say something, but that may not happen, but someone else will be saying something different. I'm sure even the WHO, when they're giving when the briefing they're coming up, and they're not sure whether they will be saying the same thing on the next day. So, I mean, we want to be optimistic, in fact. I mean, until now, our deaths are increasing, our number of cases are increasing, but I really want to interpret that increasing the cases is more, we are testing, more cases we are getting. We are not really testing a lot. And then positivity rate are very high because we are only testing the cases who are symptomatic. So, and more cases we are testing, more and more cases we are confirming, in fact. So that's one. And accordingly also, similarly, I believe that we were missing maybe some of the deaths in the past. And now you can see even Professor Anisuzzaman, sir, I mean, after his death, though he did not have the clinical symptoms of coronavirus, but he was being tested before, his funeral and everything. But then he turned to be positive also. He was carrying that. So that's the way we are more and more becoming careful to ensure that to exclude that it is not a case of coronavirus case, because we are considering each and every one as a coronavirus positive case, even when the person is died. So those are important things. So altogether, definitely the cases are more, but I believe the problem with the population as well. I mean, let me go to beyond this basic science. Consider how Bangladesh is doing. Look at Bangladesh television channels. They are really investing a lot, and the celebrities are really spending a lot of time. But still, we could not really manage to keep the people to be disciplined and to really remain at home or to maintaining the distance. I really do not know how, I mean, what else can be done. So, so it all depends on something else maybe to flatten this curve. So let's hope that something will be happening, and then, or I don't know. I mean, people are also expecting that there will be some hard immunity, but I really also do not know at what cost. So that's very important. So altogether, we just can hope, and we hope that something will be going down maybe in the coming month. But that coming month should not be months. That should be maybe next month.
Now, I would like to know what is the situation of Bangladesh in contrast to the global scenario in terms of our testing, our logistic arrangement, medical facilities, everything. How optimistic we could be?
Sure, if I may start. The entire world is going through a really, really difficult situation. It's not like some countries are doing amazingly great. Yeah, sure, some countries have been able to flatten the curve, and some countries have seen no new cases in the last week or so. However, it's hard to compare because the baseline is so different in every country. We have seen Bangladesh started with almost no testing capacity and only one lab testing. I would say we have come a long way from there. Many labs have joined, infrastructure is being built, capacity is being built. We are seeing more groups getting together to ensure that quality is assured of the sample collection and testing. So I'll say we are going in the right direction, while some might contend that perhaps not in the fastest speed. But I mean, as Bangladesh, I think we are trying. But what is very important for Bangladeshis right now is to follow guidelines, personal guidelines, because every citizen of Bangladesh needs to understand that no one is safe till everyone is safe. So each one of us has to ensure that we don't go out and maintain social distancing unless we really can't. We have to wash our hands, and we know we have been hearing washing hands from the very beginning, and we are kind of like getting used to it and kind of forgetting, but reminding people, wash your hands and wash for 20 seconds. And there's a specific reason 20 seconds is told, because it takes 20 seconds for soap to break that virus. Like getting those message across. If you don't have soap, use hand sanitizer, wear masks, and don't throw masks anywhere on the street. That doesn't help the country. If you wear gloves, don't throw the gloves anywhere on the street. I think those are personal responsibilities, ethical responsibilities that we must take on in order to ensure that the entire country is safe. And I'm not only protecting myself, but I think we are learning and going forward.
Thank you all for joining us. We hope Dr. Sejati will be with us very soon in this situation to help more sequencing from Bangladeshi samples. And we will have a very positive scenario here in Bangladesh. Thank you.
Thank you.
Thank you so much. Thank you.